Okay, and great thanks to Patrick for providing that. And as I say, although he's not able to be here for all sorts of very good reasons, just to point out, as Fran put in the chat, I think a bit earlier, he's very happy to take any questions by email. So if you want to send in an email message, that will be great. Just to follow up one question actually that I should have made from Sophia. Is now the proud owner of his own YouTube channel, where, for example, where these pub MLST events, recording to pub MLST events as videos are deposited. So yeah, this, I think it's right to say that all of the speakers have given their permission for these presentations to go on that, and they will be going up in due course, along with the full-length recording of this. So there is. That these will be available for anybody who joined late or couldn't make it. I can see there's quite a lot of chat from Arnu and a number of others in the about the issue of antimicrobial resistance, and I think this is a big question that we do have to be very aware of in this massive population of Campylobacters that we were talking about. So we do have a few more minutes for general questions. Is there anybody got something that they particularly would like to bring up? We had some good chats just starting from Sam and Noel before then. Sam, do you
yeah. So. All right, so I'll sort of carry on. I mean, again, it's a question rather than a sort of comment, but does anybody think we will be able to tell if antimicrobial resistance arose in an animal or in a human? And if so, how are we going to work that out?
Well, I think that's a very good question. Has anybody? I mean, coming along those samples, just so Rick's just said before, in the US we raise about 9 times 10 to the 9 broilers a year. If half of them has 10 to the 9 generations, a lot of Campylobacter in their lifetime. Yeah. So I think, yeah, I mean we're pretty certain, I think, that there's been a lot of fluoroquinone resistance that has arisen in chicken. I think that, because some of these are associated with chicken lineages, but yeah, I mean, of course, you can't necessarily replay the tape of evolution, can you? Noel, did you have something to?
No, but I suppose in broad terms, we eat a lot of chickens and they don't eat that many of us. So it's probably fair to say, and we're not a reservoir for Campylobacter, so I presume the large bulk of Campylobacter is perhaps a really good organism, which you might be quite so certain about in some other areas where humans are an E. coli reservoir, as are cattle. But for Campylobacter, we're a recipient.
Yeah. So, and I mean, we do need to think, I think, about sampling frames as well, don't we? I mean, not just I mean, I know, Sam, you're interested. We've done some work, and Noel as well, over the past, of sampling in low middle income countries, particularly Africa, and that really we know not very much about what's going on there.
Yeah, I mean, I think Ben Pascoe's on here, and Evangelos as well, I think.
Yeah, yes, from Peru, yes.
Well, Ben has done some interesting work on Peruvian Campylobacter isolates from children. And we did find, or he found, a lot of the usual suspect sort of chicken-associated Campylobacter, but there were also a bunch of strains of where we don't really know anything about them. And I suppose, I mean, I don't want to talk too much because there's loads of people here, but if we get to know certain lineages, and we sort of get to know, obviously from PubMLST and from the database, we get to know that if we find an ST257 complex isolate, then we start to think chicken, but there are a whole load of strains that that don't ring any bells in my head, and I know quite a bit, and we don't know what their primary host is. Like Rick's, presumably contaminants in water we don't imagine that they're growing there, do we? We think they came from somewhere else, you know, and we don't necessarily know where. Lots of
Sam, presumably for your Peruvian ones that you're finding, particularly of the children, your hypothesis includes that they've come from some other animal source, or that there is indeed a human cycle.
Definitely, yeah. And we've done a little bit in Africa, Noel, or we're trying to, you know. But I mean, and it's ideas that I learned from you and Martin, you know, 100 years ago, and that is that obviously in low and middle income settings, there could be human to human transmission networks where there's sufficient contamination of perhaps drinking water or whatever with human feces that you end up with a human transmission network.
And human adaptation.
Yeah. And so that's very interesting. Another one. Well, okay, I'm on a roll.
Okay, sorry.
Oh, go on, Arno. You say
can I make a comment on
just to say to anybody who wants to join in, if you want to put your camera on or type a comment, I mean, just to follow on, there was Nicholas Beller just said, do we know how many Campylobacter diseases are around and if trends come up? And of course, yeah, we again, this is an issue where we don't really necessarily know what's going on in some other countries. I mean, we know about the intense disease in high income countries generally, but quite what the pathogenic relationship is in some of these other low middle income countries is an interesting setting. Sorry, Arno, you carry on.
Just a quick comment on what Rick wrote about the mutations. We got to remember that the quinolone resistance is mutation based. Yeah. Primarily, but all the other resistances like the aminoglycosides, tetracycline, and are primarily based on resistance genes coming in from elsewhere, probably, or have become house trained in the Campy populations. So they're more likely to come from animal sources, those, especially the aminoglycosides. I would not be surprised if you can say that. Whereas the macrolide ones and the quinolones one, because they're mutation based, they can more be generated de novo.
Yes.
Or transformation, a natural transformation, and homologous recombination.
Yes. So you've got your camera on, Catherine. Did you have any of this? Or I mean, obviously, I think it's very interesting how you've done that combination of. I mean, we always used to try just doing flour and pour, but the combination, putting in the amino acid, the antibiotic resistance is interesting to see. There, the way the antimicrobial resistance is also a genomes. It's because you wouldn't necessarily think that. You'd think selection would trump it all, wouldn't you? But we see this as well. Certain clonal complexes have certain likelihoods of being involved in.
But I'm asking if there is a benefit for the bacteria to have this mutation of the inner G rays, because even without pressure, there is a persistence of this mutation. It's a question.
Yeah. Well, I think there is some evidence, isn't there, that they might persist rather more than you'd expect, because the fluoroquinolone resistance is supposed to be taken off, but it's still persisting very strongly, isn't it? I mean, no, we see a lot of, particularly the fluoroquinolone resistance, hanging around.
Yeah, and I think there's a PhD student along with ANSES and the APHA, Taylor Hanford, who's currently looking at doing some work trying to work out, is there a cost to the mutation or is there a benefit from the mutation, and planning to get some isolates and run them with Isabelle Kempf and Cattell in ANSES in France to look at those questions if you're actually seeing evolutionary advantage with and without. So, yeah, I think some papers in the past have gone in either direction, better survival with the GRA mutated form and better survival with the wild form.
Yes. Yeah, so just picking up some of the things from in here, there's an interesting question from Cecilia Jones about transport crates to and from farms and abattoirs have been spreading Campy in Sweden. And I mean, I think that's absolutely, clearly the place I've always thought for many years is that the chickens are most likely to get Campylobacter from other chickens. And of course, transport crates is just one way in which that can happen. And somebody else is Yetageli, I hope I've got that right, about the other species. Well, other species do cause disease, but they can be quite different. And I mean, Rick mentioned the doylii, which is a distinct group within ecology. So other Campylobacter species are important, though it must be said they're not quite so. They don't come. And then we'll just Mustafa's talking about, and this is interesting, comes in about core genome MLST and the robustness of different typing schemes. Well, generally, of course, and Ace picked this up and Catherine answered about it. Of course, whether you use a nucleotide-based scheme, well, or an allele-based scheme, of course, the alleles have the nucleotide sequences in them. So, I mean, in my experience, most of this is the level of detail. As Catherine showed quite nicely, most of these schemes are fairly robust and consistent, and it's really just the number of loci you use. So as you increase the number of loci, you generally increase the robustness and the resolution. The one thing I would say, and I would say this, wouldn't I, because we developed MLST with this in mind, is you have to be very careful with nucleotide-based schemes because they are based on nucleotides, and you have to take proper account of recombination there, which can, horizontal genetic transfer can change that quite a lot. So, and then finally, saying about the, I will expose everybody to my prejudices, but others can pitch in as well if you want. A naive question about what is appropriate to term isolate and then strain and clone and meroclone. Well, an isolate I always like to refer to is actually when you isolate something. It's a physical thing that grows on a plate. Unfortunately, taxonomy to call some strains, which is a bit confusing, but that's what I like to call an isolate, which is an isolate. Whereas a strain can be used to define, as Catherine was, a combination of organisms that that have a shared characteristics that you can see spreading. So a particular strain of Campylobacter have many isolates of them over time, and obviously clones are again a plate item. If you clone something, you get an identical organism, but most of these strains, I think, are basically meroclones. In other words, they because whenever you sequence them, there'll be variation in them. They'll never be identical, will they, Sam? Even if they're closely related. So if you think of clouds of variation, but the extraordinary thing is how stable they are, given the variation and over lengths of time, and probably only change when the ecology changes, like with Sam, rather nice demonstration of the age of the bovine variant, going back some time, at least a few hundred years rather than millennia. Yeah.
I had another sort of thought, and it sort of relates to something I've been doing. I mean, it's just a conversation again. Is how many isolates do you think we need to sample to get a reflection of diversity within a given host? I mean, that's an open question. So obviously we've got, I don't know, 10,000 from chickens now, maybe. Is that enough, or do we need more?
Yeah. Well, I think I think it depends on the environment, because it'd be interesting to know whether there's a geographical distribution. But I suspect we're getting a pretty good idea now. Personally, I think the databases are particularly large. That there aren't many surprises. There are some, but and most of the clonal complex names still have fairly low ST numbers. Interestingly, which means that they were defined early on in the numbers are arbitrary, of course, other than they're issued over time. So interestingly, in Meninge, for example, we recently had in Africa the evidence of a 10,215 clonal complex, which is unusual. Most of the clonal complexes have been named by much lower numbers, so that may have arisen recently. So, but this is something that we, by collecting the big databases, cataloguing them, we should get a bit of an idea about. I'm trying to work my way through the questions here. Sorry, Sam, did that make sense? Or
yeah, no, definitely.
Yeah, Rick's saying he didn't find any C. coli in his river isolates, which I think probably that fits some of the rest of us. I mean, Noel, you did some water exposure studies, didn't you, with your PhD? And I don't think there anybody got C. coli from exposure to water, although fishermen and wild swimmers did get C. jejuni, if I remember correctly.
Absolutely, wild bird types of C. jejuni. Yeah. But the water, I suppose, Sam has done stuff in water, and people in Manchester did water, and when they found C. coli in the water, in their case, it was these other clades of C. coli that are not the human infecting clades. In the sense of sort of three big subspecies. Sam is probably more directly involved in those water things than I ever was. Yeah.
Yes.
Yeah. That's right. So, but we found the sort of what we call the environmental C. coli clades in sort of wild waters or sort of ducks occasionally and things like that. Arnu did a big sampling of that as well and found that. But I think Jejenai still dominates. But it's an open question. Who knows? Who knows what's going on? I was just going to put one in the chat. I'm going to say it anyway. I wonder if anybody's ever looked in fish. I mean, you never know.
Well, you found them in seals, didn't you? Those were the human ones, weren't they?
Yeah.
ST21 in seals, wasn't it?
That's right. Yeah. We found them in seals, but then of course the seal is a mammal, isn't it? I just wonder if fish represent, or don't know. Anyway, be interesting.
There's a comment from Jose Alexander saying Ching Zhang's group in Iowa, some work on fitness benefits. So if anybody wants to follow that up, it's in the chat. Transmission between poultry and wild birds, Fran, from Nicholas Bellinger. Do you want to pick that one up?
Well, there's lots and lots of groups that have looked specifically at wild birds, and they seem to have very distinct populations of Campylobacter, I think related to the way they feed or their sort of feeding guilds.
You tried very hard to find transmission from wild birds on the Whiteham farm, and
yes, I was thinking most of the work that's been done is probably looking at commercially housed birds, but we started our work with the free-range birds, and then specifically because they represent the worst case scenario. So we're quite convinced that it's not coming from wild birds, but there's a comment from Arnud there saying that, isolates are still underrepresented, which they would be on the database because it's dominated probably by sort of Campylobacters from humans and chickens, isn't it? But when we've looked in other groups, we haven't seen evidence of that. And we'd go along with what Martin said about we're predominantly seeing chickens having chicken types, and then the most likely place they're getting them from is from other chickens, we would think.
Yeah, it doesn't mean it's impossible, but I mean, when we were amazed, weren't we, Fran, 20 years ago, because you'd see starlings all over and geese all over the farm in Whiteham, and feeding on the feed piles that are used for the chickens and all sorts of stuff, and we didn't pick up any really crossover at all, did you? Extraordinary.
Yeah, they would have the same types. Starlings have the same types as those in New Zealand.
Yeah, yes, exactly. The starlings look like starlings in New Zealand, but not like the chickens on the same farm in Oxford.
Do you really have wild birds in the UK, would you say so? Because the reason I said made my comment is those UOH, the University of Helsinki isolates, which are really sort of outside every of the MLST. They're really at the root of every MLST tree outside the clonal complexes. Maybe in Finland they have a bit more wild nature that you can sample from.
Well, yeah, no, that's true, but it does seem birds. I mean, the other thing that we found, we haven't published this, have we, Noel? But the interesting thing, if you look at rats on farms, that rats get chicken types, but chickens don't seem to get rat types, which is sort of quite interesting. Even though the rats have got lots of campy, so you will find the chicken types in. I think that's right, wasn't it, Noel? But you wouldn't.
I suppose rats and seagulls, to my mind. Seagulls have their own host-specific type, but they also seem to graze on everything. And rats look as though they've probably got a rat type, but also
they get stuff from elsewhere. Yeah. It's great. There's loads of stuff. Keep it coming, folks. I hope I'm not missing anything. If anybody sees anything that I haven't picked up, Was that a question? Or has it unmiked? I think that's somebody who just unmiked. Are the Campion fish, Sam said. Could message genomics approach? Well, I think, the problem with the metagenome, and I don't know if there's anybody else would like to comment about that, but the bits we've done is it's not always defined, Campy, is it, Fran? Because they're not very much even of the chicken, and even in an infected human, they're not much of the microbiome.
Yeah, we've tried, I think Claire did something, didn't she, with PHE?
Yes. She couldn't even find it in some people they knew had Campylobacter.
Yeah.
I don't know if anybody's online who wants to. But in chickens, even, it's not always very high numbers, is it?
No, when we've done 16S, we've had to use very, very high sequencing depth, and we might find, less than 1% of the microbiome is Campy, and that's in birds that we can culture and we know have Campylobacter. So yeah, it's difficult. Yeah.
Okay. Well, there's a very nice comment here from Sophia, which I think is a nice comment, and I think we will do that. And I think it said I joined late. If you guys could did not do so already, could we please have a moment of silence for Ali? I think that is a nice idea, and maybe we'll do that right at the end. We'll be closing shortly, I think. So I think he agrees. I think that will be a nice thing to do to end on. Hannah, are you online? Do you have a picture of Ali? I'm just asking Hannah, who's running the. Or Andrea, are you online? Because we did have.
Yes, I can send a single picture to.
So what I suggest,
Hannah quickly, because there's a picture on the program.
So I suggest it's a very nice thought, and I think that's a good idea. So when we close, I think we'll put that picture up for a few minutes at the end, and we can all have a few moments, and then we'll close without any further comment. Does that sound reasonable? I think that's a very nice suggestion. Thank you very much, Sophia.
Would you like any more reflections on Ali, Martin?
Well, I think let me just go through to the bottom of this, and then I would be very happy for people to do that, and then we'll go to closure. If people are happy with that, that's fine. So if anybody wants to say anything, you just put a note in the chat, and then I'll get back to it. So there's somebody here saying, sorry, I'm going to. There's some comments here from Aldert Zoma showing, if anybody wants to, showing a nice comparison to human and C. coli isolates. And Karina, hello Karina, nice to hear from you, who's got into Campy, having left the lab, which is rather fun. She used to work with us on Nicea, of course. We get STX tech runoff from cattle. A bit of a shout out, she just published a very interesting paper, Karina, on their surveillance in Ireland. We get S-Tech runoff from cattle into streams and on recreational beaches in Ireland, so presume Campy there too. Although linking in with the comment that Rick said about the sensitivity of Campy to probably the amount of sunlight probably affects how much you get environmental contamination of that type. Interesting to so Nick Bellinger, interesting to see salmon farms. Yes, I really have no idea about Campylobacter in salmon farms. And a couple of comments for papers. Given that now from Peter Woos, given that general population is highly recombinant, which of course it is there any method we can use to minimize the impact of recombination with any global population using MLST or CGMNA? But well, I think there's a number of population approaches. Sam may want to make a comment on Noel too. I think the best way of doing this at the moment is clonal frame ML would be from Xavier Didelot would be my method of choice for doing that. I don't know, Sam or Noel, whether you have anything comment to say about that.
For trees, are we talking about there, Martin?
Well, we're just looking at the impact of recombination when analysing.
Oh yeah, clonal frame ML, but the other one, of course, and I'm sort of jumping in front of Noel there, but I suspect he'd say the same thing. The other one you can use is Gubbins, which does a similar sort of thing. It identifies SNPs that are close together in the genome and therefore infers that they came in a single recombination event rather than point mutation. And Gubbins is sometimes a bit easier to run on large data sets, is it? I don't know if that's still true.
Yeah, off you go.
The answers that Ed Tabata and Arno given too, that to an extent MLST-based analysis, whether it's to an extent inherently.
Which is why MLST was invented, he says. I mean, that's true. That does help, although it's a sort of informal way of doing it. My word, this is wonderful. Thank you, everybody. It's such a. I know we've gone horribly over time, but I think this is great. The group is found Campy is low abundant found in the samples of human infection. Yeah, the similar result from Jose Alexander Rodriguez. Our group has found that Campy is in really low abundance. Yeah, which is does make it more difficult. Number of alleles, the impact of recombination. Yes, from as you said, for Campylobacter metagenomes. From Nicole, this is great discussion. Thank you. I hope I've not missed anything. Yes. CFML from Evangelos, Euclid Mars, those highly recombinant regions. Yes, back in the region. Yeah, there's a number of ways of doing it. Actually, to be fair, I think, the recombination is relative. It is not too much of an issue between Campylobacter in different lineages. I mean, they come in, and it's important of AMR, but it doesn't. I think it's the impact of recombination within the clonal complexes, particularly some of the large ones, which I think is a bit problematic. So I think sometimes subsetting your studies down and don't try and do everything at once, particularly with the clonal ML analysis. You have to ask your question: are you looking for recombination between groups or within them? Great, that's great. Is there any of our faculty from the speakers who feel they would like to make any more comments? I think that will be the only thing I'll take now after this. From Rick or Catherine, that you'd like to say further to do with any of the discussion? That's great. Andrea, do you know if Hannah's got the picture?
Yeah, Hannah has got the picture, and I would like to mention. I would also like to mention that it would be Ali's birthday tomorrow on the 30th of March.
So that was yes. So
and Hannah will share the picture in a moment.
So I think would you like? Yeah, so some really lovely comments from Karina. Thank you, Karina, in the chat about that. Yeah, so Sam, if you would like to say a few, and then just before you do that, I'd just like to thank everybody for participating in this meeting. I mean, we had a really good attendance, and a lot of people have stayed to right to the end, even though we've gone a little bit over time, which is great. I do want to especially make a big thanks to Fran Collis, who really put an enormous amount of work into organising this, and thank you to her and to all our speakers again for coming and joining in making this such a stimulating, interesting meeting. We will, as I say, put this up online, and there's lots of interesting stuff in the chat. There will be more of these events in the future. We wanted to do one on Campy fairly early on, and we wanted to do it also with the opportunity of remembering Ali. I think as he worked with her quite a while, I will actually end my contributions meeting here with my thanks to you all. I will ask Sam to say a few words, which I think is entirely appropriate, given that he worked very closely while he was here with Ali. If Hannah, you could put a picture then up for a couple of minutes, and then I think we will all leave with our memories of Ali. So, Sam.
Okay, well, so obviously it's a great pleasure to have the opportunity to sort of remember Ali and celebrate all the great work that she was involved with. I just thought I'd say a little anecdote as somebody who knew her for a long time, and obviously now I'm quite old and I've got a good job and all that stuff. But when I first started in Martin's group, I came from a sort of slightly different discipline, and I was very nervous, and I moved from Cardiff University to the mighty Oxford, and of course I was intimidated by that. And I remember Ali, well, I remember obviously I was welcomed by everybody, but I remember particularly Ali and Fran actually both telling me what to do in the lab, you know, and I also remember actually Ali being quite firm with me, and I suppose my, you know, the honest truth about it is that in some ways she gave me these harsh sort of teaching me, you know, I've got to clean my workbench space. I've got to make sure that I'm rigorous with my analysis. I've got to make sure that I get my reagents right, you know, and well, those were just enormously valuable lessons for me and turned me from a mediocre, you know, early career person into somebody, well, with at least some confidence about my ability. And obviously I went on to publish some good stuff with Ali and with other people. So that's my anecdote. It's not terribly interesting, but I hope it articulates how grateful I am. Yeah, I'm sure that Ali had the same effect on lots of people. So, you know, I'll end there.